For the third installment of our series on the cell, we are going to talk about eukaryotic cells exclusively. Remember, these U means true, and karyote has to do with the nucleus. So actually it means kernel, but this current, I guess someone who first looked at this thought this looked like sort of a kernel within a cell. So a eukaryotic cell is one that has a nucleus inside it where the DNA is found. The area that we're going to be focused on, though, is not the nucleus, but all that material that you see outside the nucleus, and all these things which we're calling, these are membranes that are outside the nucleus, and this is called the endomembrane system. So the endomembrane system is going to be this whole series of internal membranes and organelles that are within that, that together work to do something with proteins in particular, and they modify and package and transport both lipids and proteins. So it's going to include several parts. It's going to include the nuclear envelope, that membrane that surrounds the nucleus. It's going to include things called lysosomes, little bubble-like structures called vesicles, the endoplasmic reticulum, or the ER for short, and something called the Golgi Apparatus. And finally, the plasma membrane, which is that very outer membrane that surrounds all cells. So let's start with lysosomes. If you think of the word Lysol, which is a household cleaner, the way lysosomes function is to destroy and break down things. And so lysosomes are structures that have a membrane around them, so you can kind of see these little circular structures in here, which contain very powerful enzymes that digest materials that come in. So this could be digesting like larger macromolecules and breaking them down, or it could be breaking down bacteria or other invaders that could go into the cell. So it can act as a defense structure as well as a sort of a garbage structure too, just to get rid of waste products. And it even can destroy cell parts that get worn out. So we'll look at that a little bit more later. Sometimes the lysosomes, I used to refer to them as the suicide sacs of the cell, because they actually can serve at the end to recycle a cell that is dying. The biggest, most obvious thing, though, when we look at the endomembrane system is this thing called the again within, plasmic, meaning the cytoplasm, reticulum, or ER for short. Endo within, plasmic has to do with cytoplasm, and reticulum is, if something's reticulated, it's kind of like weaves back and forth. So this is made up of a bunch of membranes that kind of weave back and forth. They form these tubules and sacs, and what their main function is to do things with proteins and lipids. Now there's two types of ER. One has lots of little dots on it, which is what we call the rough ER. Those dots happen to be ribosomes. And the rough ER is involved with a lot to do with modifying proteins in particular. Because remember, ribosomes are where proteins are made. The primary structure of proteins are made in the ribosomes. But then the smooth ER, which doesn't have ribosomes, is involved with lipid synthesis. In other words, it makes lipids. Now it talks a little bit more about how we can look at it structurally. This thing within the ER, there's actually little spaces called the lumen, or this is cisternal space. Don't worry so much about that. It's just to kind of identify where we are when we're looking at these structures. But the ER is continuous with the nuclear envelope. So here in the purple is the nucleus, and surrounding it is a nuclear envelope, and the ER just kind of keeps going from there. So it's membranes again, just kind of weaving back and forth here as it goes out into the cytoplasm. Now the rough ER, we said, was involved with proteins. That's where proteins are made and modified. It's where, just like we learned with protein folding, how that all happens, it's going to happen there in the rough ER, in those spaces within the rough ER. Once those proteins become modified, those modified proteins are either taken up by certain membranes already there in the cell, or they could actually be transported out. They could be secreted out from the cell. Things like hormones, for example, or some enzymes are going to be transported from the cell. So it's made in the cell, by the ribosomes in the rough ER, and then that material, those proteins, are sent somewhere else. Now the rough ER, you can see is, so RER is short for rough ER. And it's also involved with making these things we know of phospholipids. And phospholipids are what make up all these membranes. So this is where the phospholipids, or some of the proteins, are made and modified, and then they're going to get transported to where they need to go. So we're going to take a look at transport in a minute. The part of the ER that doesn't have ribosomes is called smooth ER, or SER. And you just looking at it, you'll be able to see the difference. This picture kind of shows it to you, although it makes them look very different here. But here's the rough with the ribosomes, and here's the smooth without. Now the smooth ER does a lot of different functions. It will take some of the things, the macromolecules that were broken down, and then rebuild them into new carbohydrates or lipids or hormones. The other thing it does, though, is really important, is it gets rid of poisons. So when you take medication, some of those medications aren't good for certain parts of it. You've heard of all the side effects they talk about on TV. Sometimes even medications need to be broken down and detoxified so they don't hurt us. So detoxification of medications and poisons, and cells, especially cells within the liver, are involved with this. And the smooth ER also stores calcium. Calcium, you're going to see, has a really important role in a lot of things, including the nervous system. But we'll take a look at this later on. So just tuck that away in your memory banks. So keep going. So here's all the ER, smooth and rough, and then we go out to another series of membranes that kind of look like a set of flattened pancakes, and that's called the Golgi Apparatus. I think of the Golgi Apparatus like UPS, the packaging and shipping company, because that's what it does. The Golgi Apparatus basically takes what the ER has done, and if something needs to get modified a little bit more, in other words, you might need to add something on it or change it a bit, the Golgi Apparatus can do that, and then it's going to move the material into little trucks, which are these little vesicles. So let's say these are proteins or some kind of a hormone. That's where I'm circling right here. So the Golgi will create these little membrane buds that eventually are going to be able to leave the cell. They can enter or go through the membrane of the plasma membrane of the cell. There's actually a receiving end, and there is a shipping end. So the receiving side is the cis phase. Remember cis and trans? We looked at these when we were looking back in biochemistry at some of the isomers. If you remember those, the ones that had the double bonds had a cis formation and a trans. So we can just use it as sort of a location thing. So cis is where the materials come from the ER to the Golgi. The opposite side is trans. And so you might think of trans as kind of like the transport side. And so it's there that we're gonna get some materials coming from the ER. It's gonna go to the cis part of the Golgi, the receiving end. Goes into the Golgi apparatus. Things get modified. Kind of goes through them. And then finally, when they're done, which may involve putting a little bit of a sugar. Set a little chain of sugar molecules, which I would call oligo, meaning few, saccharides, meaning sugars, right? So we've talked about monosaccharides, disaccharides, polysaccharides, but if it's a little short chain of sugars, we can say oligosaccharides. Okay, so that happens in the Golgi. We'll look at this again with some specifics later. This is just introducing you. Okay, So now we've talked about the endomembrane system, but some other parts within the cell are also very involved with structure, keeping the shape of the cell, and also acting to provide ways for things within the cell to move around, kind of like a freeway. So the cytoskeleton is again made up of proteins, and it does a lot of things, including what you see right here. The three main parts of the cytoskeleton are microfilaments, and they show you the picture over here, microfilaments. Ones that are a little bigger, the intermediate filaments, so you can see that they look a little bigger in this picture. And then things called microtubules, which, as you can see, they're showing you here, looks like a kind of a straw, which is a tube. And each one of them is a little bit different in size and shape and different functions. So microfilaments are the tent poles. And if you've ever been camping and you've done one of those pop-up tents, then microfilaments are kind of like those things that hold the tent, together from the inside. So they're going to kind of form the shape of it, and they're made of a material called actin, which happens to be the same kind of material that's in your muscles as well. Microtubules are also form this kind of internal skeleton for some cells, and it also provides an area for things to move through the cell. We're going to look at a video that Harvard, it's a little animated version of the cell by Harvard University, and I'll point out what motor proteins actually are. So you don't need to know that they're tubulin dimers. Don't worry about that. It's just kind of describing their shape. Then there's some other little structures that you probably have seen before if you've studied cells. Very often we see these associated with little microorganisms, and these are called cilia and flagella. So these are things that microorganisms like protists, it's a group called protozoans. Some of them, you have these they're outside of the cells to help them move. Sort of like little oars, they kind of move this, or whip-like structures that move a cell through the water if that's where they're living. Now they're giving you the specifics of how their real structure is. I don't care that you know this, but it's just if you're interested, I'll tell you more. Outside the cell are also structures that are involved. In other words, outside the plasma membrane of a cell. One of those you've already learned, and that's that plants have a wall made of cellulose that surround the membrane and protect it. Also outside the membranes of cells is an area that we call the matrix. If you're into movies and you've seen the Matrix movies, is this part that's outside of the normal part of the cell, but it has all sorts of stuff in it. And I'll point it out in the next slide. But you've heard of collagen and other types of proteins. And we're going to see some of the parts of this matrix here in a sec. So if this is the cell, so notice this is the phospholipid bilayer. This is the plasma membrane. And this is the inside where my cursor is of the cell. This is the outside of this cell. And see all this, it looks busy out there. So here's in some cells, for example, skin cells, you might find a lot of these collagen fibers. And there's some other things here, like here's polysaccharide, and here's little proteins, and this whole thing which is called a proteoglycan. Do not need to worry about that. Do not freak out at that word. But it's just to show you that some of these have other names. Last but not least, cells don't usually operate by themselves. They usually talk to each other. They connect and they communicate. So there are junctions that communicate information between cells that are next to each other. And plants and animals have different types. So we'll take a look at just a couple. In a plant, there are little channels. So here, if this is one plant cell and this is another plant cell, there's a little channel. It's like a door between these two rooms called a Plasmodesmata. And this is misspelled, by the way. The word at the top is correct. And it's just a little connector that allows information to go through.